In this lecture, we will discuss about proteins purification and sequencing. So, the use of different processes to isolate one or a few proteins from a complex mixture, usually cells, tissues, or whole organism, is known as proteins purifications. Protein purification is a vital process for characterization of function, structure, and interaction of the protein of interest. So, due to protein purification, we characterize the function, structure, and interaction of proteins. The methods used during these processes will depend on the nature of both proteins and the solutions. The separation steps usually exploit differences in protein size, physico-chemical properties, binding affinity, and biological activities, and the pair results is usually termed as protein isolates. So separation steps uh, take into consideration these types of parameters, and the pair results is usually termed as protein isolates. The protein purification is either preparative or analytical. The preparative purifications aims to produce a relatively large quantity of purified proteins for subsequent use, such as preparation of commercial products such as enzyme and nutritional proteins. So preparatory purification processes produces large quantity of purified proteins. The next is analytical purification. It produces relatively small amount of protein for a variety of research and analytical purposes such as identification, quantification, and studies of protein structure, post-translational modification, and its functions. Next is the methods involved in purification of protein. We will discuss this a general procedure of purification method. First is chromatography methods. These methods exploit the physical properties of either the target protein or other elements in the solutions. So this is the basic aim of the chromatography methods to exploit the physical property of the target proteins or the other elements in the solutions. And we use various types of paper, gas, and liquid chromatography methods, and the proteins and surrounding elements are typically dissolved in a mixture and then passed through a stationary phase prior to the analysis. So for purification purposes, we use different types of paper, gas, and liquid chromatography methods, and proteins and its surrounding elements are first dissolved in a mixture and then passed through a stationary phase used in the chromatographic techniques before the analysis of protein. The next step after the chromatographic techniques is the centrifugation, filtration, and sonication, and other fractionation techniques used to break up, remove cells, part that surrounds and contain targets, protein like cell membranes and DNA. So these are the fractionation techniques in which is used to break and remove cell parts that contains or surround the proteins. For example, if protein is present in cell membrane or in DNA, so are mm, these uh, these organelles are fractionated. So in order to break it and remove the cell part. The next is the precipitation. During precipitation techniques, ammonium sulfate is added to the solution and protein samples are gathered concentrated as they form a precipitate, and this precipitation step is often used for the bulk protein extraction, the protein extracted in large quantity. The next step is the gel electrophoresis technique. It is used to identify denatured proteins by passing the samples through an electrically charged gel matrix. So this is also an important technique used for the purification process. The matrix typically separates proteins molecule by pH or molecular weights. The most important types of gel electrophoresis technique is the sodium dodecyl sulfate polyacrylamide gel electrophoresis, also known as SDS page. This is the most important technique used for the purification of proteins. Due to the complex nature of proteomic pool, purification of protein routinely involves more than one of the above techniques. So, as we know that proteins are complex in nature and the purification usually involve more than the above techniques we discuss. Next is the protein sequencing. The process of determining amino acid sequence of all or part of the protein or peptide is known as protein sequences. Most proteins are much too large to sequence directly, so a small peptide fragments are produced, and for this two general methods are used to produce peptide fragments. First is endopeptidases or protease are those enzymes that cleave peptide bonds between amino acid and within the polypeptide. So endopeptidase or protease cleave peptide bonds between amino acid and within the polypeptide chain. And next one is the exopeptidase. It is used which are specific for one end or the other. So it cleaves the ends of amino acid or polypeptide, either aminopeptidases or carboxypeptidases. The amino end or the carboxy end. But the important one are endopeptidases. These are most useful as it is specific and cleaves peptide bonds at certain location in the chain. For example, C-terminal side of the certain amino acids. The sequencing of the cleaved proteins fragments are done by the two important methods are, first one is Edman degradation. It is very important reaction for protein sequencing. Here the amino terminal residues is labeled and cleaved from the peptide without disrupting the peptide bonds between other amino acid residues. So in Edman degradation, the amino terminal residue is labeled with a specific reagent and cleaved from the peptide without disrupting the rest of the peptides. So the steps are first adding phenyl isothiocyanate, which create a phenyl thiocarbamyl derivative with the N terminal of the proteins. So you can see here phenyl thiocyanate is added to a polypeptide. This is a polypeptide. This is the chemical. So it creates phenyl thiocarbamyl derivative. You can see here it combined with the amino terminal. You can see here it combined with the amino terminal side. Next, the N-terminal is then cleaved under acidic conditions and creating a cyclic compounds of phenyl thiohydrogenation, which is also known as PTH amino acid. You can see here after this, you can see this is cleaved under acidic conditions. So this is the rest of the polypeptide chain and this is the PTH and containing amino acid. You can see here this is easily identified. The amino acid is so this does not damage protein and leaves two constituent of the peptide. This method can be repeated for the rest of the residue, separating one residue at a time. So you can see here, this separates this amino acid residue. So you can see, and this is can be reused and attached to this polypeptide and cleave another residue. So it can be using and separating one residue at a time. But the major drawbacks of this technique is that the peptide being sequenced cannot have more than 50 or 60 residues. So it can work only on 20 to 30 residues. So this is the major drawback of this technique. Also, it proceeds from the N terminus of the proteins. So it will not work if the N terminus has been chemically modified. So this is another drawbacks, that this technique only use the N terminus of a protein, and if the N terminus is modified, so it will not work. Nowadays, automated Edmond sequences are now widespread use, and it able to sequence peptide up to approximately 50 amino acid long. So the automated Edmond sequencer are now available. The next technique is used for the sequencing is the mass spectrometry. Mass spectrometry is analytical methods that measures mass to charge ratio, the mass and charge ratio for the ions in a gas phase. So it is a very widely used methods nowadays for protein sequencing and identification. So this mass spectrometry determine the mass and charge ratio of the ion in gaseous phase. So these are all this is all about the protein purification and sequencing. Thank you.